So I'm talking today about negative and positive control on the genes in DNA. And we're specifically in prokaryotes because eukaryotes are a little more complex. But in positive control, one example is an inducible operon. Operon, of course, is the promoter area and all the genes that code for the individual types of proteins needed for one type of operon. If we have the Lac Operon, that would be the genes that would make all the proteins that would help make the enzyme to break down lactose. Okay, so any case, if we look upstream from the regulatory gene, we can see that from the upstream from the promoter region, which is the switch, the operator, and the marker on the DNA that tells where the RNA polymerase should attach. Upstream, we have the regulatory gene that is part of the switch that turns this on and off. Now, the regulatory gene produces a messenger RNA that will eventually make a repressor protein, and this repressor protein will, in fact, be able to bond or bind with the operator. And we learned that inducers, okay, can bind with this protein to change its shape. So without an inducer, this repressor protein, it's a negative control because the repressor will bind to the operator and prevent the promoter from going, prevent the RNA polymerase from going forward and transcribing the genes for the proteins that make the enzyme to break down lactose. Okay, so you can see that this is negative control because we can stop it. In diagram B, you should see if this is the Lac Operon, and this is again just one example, Lac Operon, of a negative control system. We learned about the trip operon, but in any case, same thing here upstream. We produced the messenger RNA that eventually will produce the repressor protein that, given no other binding to or no other inducers in this case, would attach to the operator like we just showed. But in this case, we have an inducer molecule. So for the Lac Operon, it'd be allolactase, which is basically a component that comes from lactose. So if you have lactose in the body or present for this cell, you'll have allolactose that comes in the same source. And this allolactose is an inducer because what it does is it binds to the repressor proteins, changes its shape in a way that it cannot bind with the operator, and it induces these genes to be transcribed and eventually made into the proteins necessary to break down the lactose. So we would say the inducer induces the system in this case, but it's still a negative approach because the switch is turned on and turned off by this negative control system that will, if the inducer wasn't there, if the lactose wasn't there, then the allolactose wouldn't be there and it wouldn't bind, and this would fit right into the operator as written and it would shut it down. So again, this is an example of an inducible system. We talked about the TRIPS, which was a makes a repressor molecule as well, and it works very similarly in that it can shut the gene on and off. Now, in a positive control system, okay, we have another mechanism by which we can enhance, and I say enhance because we can't turn on and turn off. It's important that you get that. The repressor proteins are a negative system because they can stop the operator, or at least they can bind to the operator, which would physically stop the RNA polymerase from going forward and therefore stop the gene. That's a negative control system. Well, we're talking about positive. We want to stay positive. Well, what we have is an activator protein, and this could be called CAP, okay? It's a type of protein in the Lac Operon, and what it can do, if it's activated or it's in a shape, it can actually bind on the promoter that enhances the ability of the RNA polymerase to attach. Now, so this system doesn't stop or start or shut off or turn on the gene. The gene has to be on or off based upon the repressor protein. So in this case, the repressor protein has been neutralized by the inducer. The inducer changed its shape and it can't fit like we just talked about in the Lac Operon. See, the Lac Operon has a negative control system working here. And we would say that that repressor protein made by an upstream regulatory gene has not, okay, does not attach because of, let's say, the inducer attaching to it, like we just talked about. So that this gene is in the on position, has to be on for this to even have a chance to work. But what we can do is we can enhance the ability of the RNA polymerase to attach itself to the promoter. And if we can help the RNA attach itself, then we're going to make a lot more of these genes or this enzyme or these proteins eventually than we would normally. See, RNA polymerase does attach to the promoter, but it doesn't have a high affinity. So without this system, this would go forward a little bit. We can think of the positive control as a volume control. I can turn the gene on and off with a regulatory protein like we just talked about. That's negative control. But I can increase the rate of the production of these genes and turn the volume all the way up to increase the rate in which I do so by having this activator protein attached. That's the difference here. So if we look down, we have these messenger molecules, and for the Lac Operon, that'd be this the C camp, okay, that would attach to the CAP, and when it does so, it changes its shape in a way that it now has high affinity for the promoter. When that goes, the RNA polymerase now attaches to the promoter much better, or it has now a high affinity. And by helping the RNA polymerase attach much, much easier, the rate in which we make the gene is increased. If I increase the amount of this C-camp or this messenger molecule, I will increase the rate that it activates the cap, which in fact will increase the rate that it attaches to the promoter, which will increase the rate at which RNA polymerase will attach and therefore transcribe. So the positive type of control here is that really it's an enhancer. It basically increases the rate of the on position. Remember, RNA polymerase will attach without the cap being activated, but does so at a low rate. This really, really, really helps RNA, if I say that more times, enhances its ability to attach and therefore increases the rate. Rate means how much messenger RNA I can make of this area per time, which means I increase the rate of the protein I make per time. So we have a way to not only turn the gene on, but make it go faster or even slower. And that's what we do in the Lac Operon, because the Lac Operon and the E. coli has really two competing sugars it utilizes. It can utilize glucose, and it loves glucose. And when there's a lot of glucose, why would we need to break down lactose? Even if there is a lot of lactose in the system, we prefer glucose production because it's a more efficient system. So even if there is a lot of lactose, which would do what? It would bind to the repressor protein and prevent it from attaching, keeping the operator on. But with a lot of glucose available, this would go on it would go on, but it wouldn't produce a lot of enzymes to break down lactose. It'd be on a little bit. Now, if we have low glucose, what that would do is produce a lot of this C-camp in the presence of a low amount of glucose, which means it'll bind to the cap, change its shape so it attaches, and increase the rate of lactose production. Okay, so again, if you need to see that exact way, go to your book, but positive control and negative control work hand in hand. In the case of the E. coli, we want the gene to be on, but how much it goes depends upon how much of this enhancer or activator protein is working. Hope that helped.